today are two lead scientists on the front line of coronavirus research from the Sanford Burnham Prebys Medical Discovery Institute. Dr. Chanda is the director of the Immunity and Pathogenesis program. He is a Stanford-trained scientist who previously worked at the pharmaceutical company Novartis before joining the institute in 2007. He's a renowned virologist and has spent I'm not sure how to pronounce that one, and has spent many years developing innovative approaches to treat viruses such as HIV, Ebola, dengue fever, and respiratory virus, including MERS, SARS, influenza, and now the current virus, SARS-CoV-2. Dr. Snyder is the director for the Center for Stem Cells and Regenerative Medicine. He began his career as a physician scientist at Harvard University and Boston Children's Hospital. Today, Evan practices neonatology and neurology at UC San Diego while simultaneously doing groundbreaking stem cell research at the Institute. Evan has served 2 terms as chairman of the FDA's Cellular, Tissue, and Gene Therapy Advisory Committee and has been elected to the very prestigious Association of American Physicians. We'll have the Q&A session after each of them has spoken, so please wait till both. And we'll also put the link to the Institute's website in the chat feature, so you can copy and paste that and check it out later. So without further ado, I'm going to introduce Dr. Chanda first. Let me unmute you.
Hi, Melanie.
Hello.
Thanks for having me on. So did you want me to just talk a little bit about what we're doing in the lab and as far as our COVID research goes? Yeah, so obviously this is a new field for everybody. So we got into this field probably a little bit earlier because we are virologists than most people. There's been an incredible rallying around the flag for a lot of different scientists from different disciplines, but as a virologist, we were out on the front line as SARS-CoV-2 is a coronavirus. And whenever you hear of a coronavirus or an influenza virus that's new and that's having an outbreak anywhere in the world, alarm bells start to go off. These are viruses that are considered to have high pandemic potential and are especially dangerous. So this is in comparison to things like Ebola or Zika, which again are dangerous, but for Zika, they need to be transmitted. That needs to be transmitted with a mosquito. With Ebola, it's really close bodily contact. Whereas with respiratory viruses, it's one person getting on an airplane and flying someplace to just seed a new outbreak. Okay, and so our story actually is quite interesting, and it started with a postdoc of mine who had gone back to China for the Christmas holidays, and he had emailed me and he said, "Hey, look, there's this cluster of unexplained pneumonia happening in Hubei province in China, and they've linked it to a coronavirus, and it's probably a lot worse than the reports you're getting in the media." And so given the fact that this is kind of our space, what we work on, our immediate thought went to, boy, how do we help? How do we try to help and pitch in as quickly as possible? It has been and continues to be a race around the clock. We're set up here with what's called a Biosafety Level 3 containment facility. So that allows us to work with some of the most nasty viruses in the world. These are, you've seen people with the spacesuits on. That's the folks in my lab that they have to gown in and work on that. And so we immediately jumped into this because we knew we had to do something. And the first strategy we took was to try to find new drugs for this virus. And by new drugs, it really meant trying to find old drugs that could be newly applied to SARS-CoV-2. The reason for that is that most drugs take anywhere from 10 to 15 years from inception to bringing to market, okay? And this was not the kind of time that we we had in that current situation. Now, mind you, this was at that time restricted to China, but we knew that this could be something much bigger. We were hoping it wouldn't be, but we knew that if we had gotten an early start to this, we may be able to impact it. And so what we did was we teamed up with two critical groups. One is a group at the Scripps Research Institute here in La Jolla. So they had developed, in conjunction with Bill and Melinda Gates, this amazing scientific resource where they went through and they compiled 12,000 of the drugs that have ever been in clinical trials or approved by the FDA. So they had a physical collection of 12,000 drugs, okay? And so we knew we had this asset that we can then try to test against SARS-CoV-2. However, at the time in January, that virus was not available to us in the US. It was confined to China, and getting that virus out of China was, you can imagine, just an insurmountable task. So what we did was team up with a group in Hong Kong that had isolated the virus out of a patient over there. And they were working on culturing the virus. And we were going to send one of our scientists out there to teach them how to screen 12,000 drugs. So to put that in the context, most labs screen maybe 1, 2, 5 drugs. So you really require special automation, special skill sets to be able to do that. And we happen to have that kind of skill set. So unfortunately, as our postdoc, as our scientist was ready to pack her suitcases with the machinery and this library of 12,000 drugs and fly out to Hong Kong, there was a travel ban that was put into place by the US government. So she could go, but she wouldn't be able to come back. And so again, that was not a tenable situation for us. So we did the next best thing. We essentially collaborated by iPhone. So there's an application that's analogous to WhatsApp, which is WeChat out in China. And so we downloaded WeChat, and my scientist got up every night in the middle of the night and talked through how to screen these 12,000 compounds against SARS-CoV-2. And really just an amazing effort by the group on our side and the group over in Hong Kong to be able to pull that off really by the end of February. Typically, these things take minimally 6 to 8 months to do if you are under the best of circumstances and if you are physically present. So to be able to do this from 7,000 miles away in the middle of the night, really hats off to the efforts of the people that are involved there. And so we took those results, and by March, we had gotten the virus into our lab. And so we've been growing it in this BSL-3 high containment facility. We took that data. We were able to validate about 30 of them that look really promising. And again, these are all molecules that have been in humans, so we know that they're safe, right? And now we know that they work against SARS-CoV-2, at least in the lab. Now we're going into animal studies. If we can show that they work in animal studies, then we can jump right into clinical trials to supplement things like remdesivir, which just got emergency use approval, right? We definitely need more than one of these drugs. Remdesivir is good. It's not great. It's not the magic bullet. So we're going to need a cocktail of therapies like we use for hepatitis C, like we've used for HIV. And that's really our goal. And because we're able to leverage the efforts and the investments that have been put into testing these drugs for other indications, things like Crohn's disease or things like MS, we know that we don't have to go through those safety studies. So now we can just go into patients, see if they work. And there's an incredibly compressed timeline now based on this emergency use authorization that's in place by the FDA. So we feel that if we can show if these molecules work in these animal studies, we can go into patients and have something that can supplement remdesivir or use in combination with remdesivir by what's likely to be an anticipated second wave of the virus coming out in late October. So that's a brief summary of our foray. It's still early days, but just a remarkable amount of work compressed in really adverse situations that scientists in my group and the others that we've been working with. And now we've branched out to work with folks like Evan and Carl Ware here at the Institute and Ralph Bodmer and Chris Larson, who bring other specialties to bear in our fight against this virus. So Evan will tell us about this incredible platform that he's developed that will allow us to model how this virus does in lung tissue, which will help us evaluate how good these compounds might actually be. And other folks are looking at cardiac tissue or kidney tissue or intestinal tissue or immune response to the virus. It's really been all hands on deck, not only here at the Institute, but across San Diego, where San Diego has really become an epicenter in the battle against this pandemic.
Thank you, Dr. Chanda. Dr. Snyder.
Hi, can you hear me all? Everybody can hear? Good. Well, I should say it's actually a great privilege to be coming back, so to speak, to the JCC. Both my sons went to the preschool there, and I kind of missed coming back, other than though one of them is still in the theater group. So we come back periodically when he's in a show. So this is great coming back to talk about something else, actually, for me to be in the spotlight instead of Madden Snyder. Anyway, I also interestingly have a China story, very similar to Sumit's. I was in China the second week of December, and I also started hearing about an infection that was in Wuhan province. And because I got to know a number of my hosts, I was on a speaking tour, I stayed in contact with them, but was also paying attention to what was going on. And it sounded fairly serious. And in fact, in early January, I was emailing back and forth and asking whether they were okay. And at the time, they were saying, "We're fine. It looks like it's just staying in Wuhan," which didn't seem to be accurate. So I came back already, like Sumit, keeping track of what was going on with this viral infection, which was primarily being seen as a pulmonary infection. And obviously, when it started really getting out of control and coming to this country and overwhelming the healthcare professionals, as a physician, my first impulse was not a scientific response. It was actually, I need to get on the front lines and help my colleagues. They're being overwhelmed. And then, of course, I remembered, but I'm a neonatologist, and I'm not sure if they want a newborn intensive care doctor running the ventilators for 80-year-old people, even though I will assert that I think I can run a ventilator better than a lot of the gerontologists. But what we could do, which seemed very, very useful, would be to help model what the virus was doing in the body and maybe drugs that could combat it. And I started talking to the people in my lab who were growing, and I'll show you what they are in a few minutes, mini lungs in a dish, and saying to them, I think nobody knows anything about this virus. Nobody knows what cell type it hits, what it does, what its life cycle really is, how it goes from cell to cell, whether, or even what its receptor is, even though it was starting to emerge that it's this ACE2 receptor, but it looked like it could be others as well. And then I heard that Sumit had actually, not only had he been on the case, he had this heroic effort, that he just described, and I really do mean heroic, where he had already screened 12,000 FDA-approved drugs, meaning these could be fast-tracked into patients because they already had a package of the FDA. We already knew what their doses were. We already knew what their side effects were, what their biodistribution was, what one needed to do. And Sumit and his group, as you saw, winnowed 12,000 drugs down to 30, which is astounding. And of the 30, further winnowed it down to 6 that really looked like they could be quite useful, even at doses that could be achieved in humans. But as most high throughput screening has to be, particularly for viral infection, it has to be done to be efficient in cell types that are not really representative of what COVID-19 is. Real lung cells that are developing. A lot of the cell types that Sumit has to do are human cancer lines or even monkey kidney cell lines and things of that sort. So we thought that maybe what we could do to help fast track drugs, these particular drugs into patients, would be to use an authentic mini lung system. Thinking that maybe if we could show that these drugs that had already been shown to be safe and effective in humans, maybe for another reason, but they certainly were blocking viral infection. If we could show that they actually do what we think that they should do in these mini lungs in a dish, maybe we could make an argument to the FDA to bypass at least some of the animal modeling, particularly because we're still struggling with a good animal model, and make the argument, particularly if it's been a drug that's been in lots and lots of patients, to start a clinical trial now using one of these drugs. Now, I'm going to share my screen because I want to show you what these look like and maybe what their advantages. So I'm free to share my screen. Is that true, Melanie? All right. All right. Hopefully you're seeing a picture of, well, the first thing I want to call attention to is Sandra Libel. She is a fellow neonatologist, and this is her technology in the lab. We were growing these mini lungs, and I think you can see, can you see my pointer? Hopefully you can, my cursor. You can see that you're growing these mini lungs that almost look, they have little bits of branching coming out. Here's another example. We had been studying, Sandra in particular, had been studying a genetic disease that babies get that makes their lungs act as if they're premature babies. I think most of you know that premature babies, certainly in the old days, and even now, can have lung disease because they don't make a substance called surfactant, which is almost like a detergent that makes the lungs very flexible. Premature babies have that problem, but we have ways of giving back surfactant. Sandra was looking at a disease where the babies never have the ability to make surfactant because they're missing the gene. And we were studying that. And in fact, in the course of doing that, Sandra made what essentially is the literature's complete, first and only complete mini lung in a dish, all the cell types, both proximal and distal and things of that sort. And basically the way she does that is when you look at a lung, as you can see on this next slide, it actually is made up of all different kinds of cell types in different parts of the lungs doing various kinds of things. And the air sacs are called alveoli, and they're lined by another kind of cell type that makes this surfactant. Now, very interestingly, what caught my attention was that some of the patients with COVID-19 were getting what is called adult respiratory distress syndrome, or ARDS, which is the adult version of what the babies get. They eat up their surfactant. So that caught my attention that maybe we can help model what's really going on with the disease. Now, the way the lung develops is through all these various steps to give you all the various cell lines. And it actually begins with a lung stem cell that then branches out and has offspring of all different kinds. What we do in the stem cell field is also start with a stem cell that can give rise to every part of the body, in fact, but definitely if you give it the right recipe, the recipe that nature uses to do this, you will get lungs in a dish. I just want to show you how dramatic and how robust this process is. Hopefully you're seeing a movie where Sandra has added just the very, very last steps of allowing these mini lungs to achieve a mature configuration and mature cell types. And they're so mature, in fact, and so complete that hopefully you can see this, that they have smooth muscle and actually start contracting as if they were making respiratory movements. So with that in place, we thought, well, this would be a great way to infect these, see what the virus does, and also see what the drugs do in terms of blocking that kind of virus. Now, the other thing that I think is important to recognize is that the way that we make these lungs, as I said, was from a stem cell. Now, where do you get the stem cell? Where we can get the stem cell from are from patients. Now, what does that mean, getting them from patients? We can take skin cells or blood cells and push them back in developmental time so that they act as if, even though they're coming from an adult, even though they're coming from something like skin or blood, we can push them back to act as if they're still in the embryo, still in the earliest stages of the embryo. And then, of course, then advance them forward to give you these lung cells. But what is very, very interesting and cool is that we like to say these are patient-specific mini lungs. In other words, they have the characteristics of the patients or the people, the individuals that donated these cells. So you've heard a lot that this disease shows an enormous amount of disease disparity. In other words, older patients get it worse than younger patients. African Americans and Latinos get it worse than Caucasians. Males get it worse than females. Well, these lungs can be African American mini lungs, or they can be male mini lungs. We may not be able to get to why these various patients have this increased susceptibility, though of course we're trying to do that. We're even making these mini lungs from patients that have problems with their immune system and are very susceptible to infection. We'll try to get to why there's a difference. Maybe they just, the younger just need to develop into older lungs and then they can get infected. We may not get to the heart of it, but at least what we'll be able to do is, out of Sumed's drugs, because we can test the drugs against these, maybe all we need to do to make sure that the drug works for everybody is to increase the dose or increase the duration of therapy or combine it with another kind of drug. So what can come out of this is not just increased knowledge, but maybe even better dosing and better regimes for patients that are particularly susceptible. So that's one of the goals that we'll be able to do. The other thing we can do is really, really make it act like a real lung. So many of you know that a real lung has more than just lung cells in it. It has blood vessels. It has inflammatory cells called macrophages. Well, this lung organoid can now be invested also with these kind of very nice blood vessels. You can see it over here, the lung organoid with the green blood vessels, and also with these red inflammatory cells. So now we really have a system that when it's infected with the virus, maybe we can also get the phenomenon of what's called cytokine storm. That's this overwhelming inflammatory reaction that also may be the real killer in some of these patients. Also, we can see whether the blood vessels are exceptionally leaky or exceptionally friable to give rise to what we call pulmonary edema. That's when the lungs in the patients get flooded with fluid. So we can begin to look at that. And then the last thing, of course, is that's great to say, can these guys really be infected with the virus? And thanks to Sumit, who created, he not only has the real virus, but of course, sometimes that's like throwing a hand grenade into the system. And we're doing that now. Sandra right now is in the BCL3 facility infecting these organoids with real virus. But until then, Sumit made available to something called a pseudovirus. What it does is it has everything that the virus has except the nasty bits. It binds to the cell and then it gets internalized and stops there. But we can be able to see who gets infected, who doesn't, what happens after it gets infected, and what not. And he has two flavors. One is over here. So here's a lung organoid. And here it is infected with the pseudovirus, all green. And this is just a higher version of it.
We're actually not seeing that slide. Can you move it forward maybe?
Can you see it now?
No, we're still on the original lung slide. We weren't able to see the little video you showed either.
Oh, you should have told me that. I had no idea.
I just was reading it. I'm sorry.
Oh, so you didn't see any of that stuff?
We see the
see
first
any of the
slide
slides
that says lung organoid with Sandra's face.
Oh my gosh. I showed about 5 slides. Oh, you should have told me. I didn't know.
I'm sorry.
I'm seeing it change. Okay. Can you see the movie now?
No.
Oh my gosh.
Well, we'll get that worked out.
All
There,
right.
now we see them. Now we see them, yes.
Well, I had it in presenter mode. Okay, I'm not going to I'm not going to restate the words. I'm just going to show you. I'm sorry you didn't tell me that. I would have done this. All right, so those are the lung organoids when you add and you get them to their very mature last bit. I think you can see them expanding and becoming the final stage of lung development. Here's the movie. Can you see this? This is the movie where they actually have respiratory movements, contracting and releasing. Are you able to see that? Just give me a thumbs up if you can see it. Yes? Okay. These are the blood vessels that we use to invest the organoid over here. Now, the interesting thing is the blood vessels can come from the same stem cells that we made the lung organoid. So if that's the case, we call that isogenic, meaning, so I was making the point that we can have patient-specific lung organoid, so an African-American lung organoid or a male lung organoid. Well, the vessels will come from that exact same patient because the stem cell can go down any direction. This is the lung organoid over here on the right. All this red are macrophages. In fact, they're lung macrophages that can also come from the exact same stem cell. So everything here will emulate what the lung is. And now here's the slide. Can you see this now? Here's a lung organoid that's been infected with the virus. Give me a thumbs up if you can see that. You can see that? Okay. So, and now here it's at higher power, really it's green because it's been infected with what we call the pseudovirus. It binds and then gets internalized. Now, not every cell in the organoid gets infected. And part of what we need to do is find out what cell type does the virus really target? And is it only this one receptor that everybody thinks is important? Or are there other receptors? And does that develop? And maybe kids don't get the disease as badly in the lung. They get it other places, but they don't get it as badly in the lung because they just haven't developed those receptors. But then as they get older, they do. But the kids do have that receptor on their blood vessels, and that's why they're getting this huge inflammatory reaction that's kind of like Kawasaki's disease. This is one way of looking at infection. Another is to infect them and then just have them light up. Imagine that there's a microscope that can read how green this is. Well, we have another version that Sumit made that lets them light up. So this is this is a lung organoid that's now very mature. Anything younger just doesn't do it. But when it gets to the right level of maturity, there it goes, it lights up because it's been infected. Now what we can do is take some of Sumit's drugs, test it here, and the prediction would be those drugs would block that level. So it looks just as low as that. And we could see, does the drug do it? Where does it do it? Does it work on all cell types? Does it work on the lung cell types? Does it work on the inflammatory cell types, the macrophages? Does it work on the blood vessel? Do we need to combine some of these drugs? And that's what we're doing. And our hope is that of those that Sumit discovered, and like I said, he's the man. he's the one in the world who's come up with the leading catalog of new drugs that we can fast track to patients. And if those drugs work and they look as if they're clinically relevant, we'll be talking to the FDA, in weeks or months just to see what it is we need to do to start a clinical trial that it doesn't cure the virus, but at least blunts its action depending on whether you've been exposed or not exposed, or how long ago you were exposed, or what symptoms you're experiencing. So I'll stop there, and I'll be glad to take any questions.
Thank you.
I am so sorry. I didn't realize. So I was just talking. I didn't realize that you couldn't see any of the slides I was showing.
No problem. I apologize. I didn't get to it sooner. I didn't want to interrupt you, and then I thought it was my computer being slow, and then anyway. Okay, so now it's time for some questions. So why don't we go ahead and put some questions in the chat, and I'll read them off. I'm also going to put the website to the Sanford Burnham Prebys Medical Discovery.
And I'm gonna stop sharing my screen unless people want.
Okay. And Dr. Chamda, you're unmuted as well, so you can answer questions too.
Okay.
I can go ahead and start with a question if we'd like. For those that have had COVID and are recovered, are they now susceptible to contracting the virus again? We've heard stories of some people getting the virus more than once in China. What would have caused this to occur? And is this multiple infection example unique to COVID?
I can start out. So there's two explanations for why people are getting it again. One is that they just never got rid of the virus, and the virus just flared up again. The other explanation, and what the data suggests, is that people who had mild symptoms actually haven't developed protection. And by that, I mean what are called neutralizing antibodies. So these are antibodies that are able to take out the virus. And so you may have antibodies against the virus, but they may not be effective in blocking the virus. So we're seeing, and this is early data, but we're seeing that if you've had mild symptoms or were asymptomatic, even if you test positive for an antibody, those antibodies may not actually be protective. Okay, and so this is not new to COVID. Actually, things like other coronaviruses where we get the common cold, your immune response either isn't protective or is not durable, which means that it goes away after 3 months or after 6 months. Okay, and so I think this is going to be particularly important when it comes to the vaccine. Because we need to make sure that the vaccine is eliciting what are these protective antibodies, these neutralizing antibodies, and then these neutralizing antibodies are indeed durable, which means that they need to last for 6 months to a year right? Otherwise, you're going to have to keep going in and getting boosters, which is just not feasible for an entire world population. I can't,
Thank
you're
you.
on mute. Okay, yeah,
Does anybody else have any? I'm sorry, I'm still new to Zoom myself. So
that's okay.
Get out. I have to be honest with you, this is my first time running it by myself. Usually I have
Okay,
my
you're doing
assistant
great.
with me, and so I'm trying to look at all the screens all at once. So anyway, thank you for your patience and learning. Okay, I don't see any questions in the chat, so I'll go ahead and ask one. The WHO reported this week that it is rare for those who are asymptomatic to transmit COVID to others. But we also hear that many people with COVID-19 who may appear to be asymptomatic actually could just have mild or atypical symptoms, or they could be pre-symptomatic. The US Centers for Disease Control and Prevention estimates that 40% of coronavirus transmission is occurring before people feel sick. Can you let us know what your thoughts are on these findings and what the new data means to you? And also, what are your thoughts on the accuracy of contact tracing given this new data?
I'm going to let Sumit take that.
Yeah, so the WHO is walking that statement back. You know, I think there was a disconnect between the scientific output and the press output. So the press took their statement to really mean that asymptomatic people cannot spread the virus, which is not true, and I think they've clarified that. Now, that being said, you know, if you're coughing or sneezing, you're spreading the virus a lot better than somebody who isn't. Okay, and so I think the lesson here though is that we're all learning as we're going, right? We're really building this plane while we're flying it, right? And so you have to be, you know, the people are putting out statements. And we're getting this in science too, which is something that's true this week changes in 2 weeks. Okay. And so, you know, I think that the wrong response would be, oh, the WHO doesn't know what it's talking about. I think it's a combination of new data and some mistranslation to the press that led to that kind of output. But I think it's still safe to say that pre-symptomatic or asymptomatic people are still going to be spreaders of the virus.
Thank you. We have a question in the chat. Following up on the recurrence question, would you expect a later illness similar to shingles following chickenpox?
Probably not, because that comes from a virus that remains dormant. Okay, for COVID-19, once it's cleared, it's cleared. There's no remnants of that virus left in your body for it to reappear. That doesn't necessarily mean that there may not be long-term implications that result from the virus infection based on the damage it does to the body, but that's unrelated to the physical presence of the virus. So we wouldn't expect these kind of shingles or other manifestations that are directly stemming from the flare-up of the virus, but we can and should be anticipating long-term effects of people, especially with severe viral infections, on their effects, not only on the lung, but there seems to be effects on neurological effects, cardiac effects, effects on the kidney. So this is going to be, I think, the fallout from this virus. I think we're only at the tip of the iceberg here in understanding the damage that it's doing to people especially with severe disease. Evan, did you want to add anything there?
No, I agree. You know, so we're only now beginning to appreciate, as Sumit said, not so much what the virus does staying in the body, but the residual after it of its attack. And I'm particularly thinking now that we're starting to learn for still somewhat obscure reasons, that one of the effects of the virus or a secondary effect of the virus, so maybe not directly caused by the virus, but as a result downstream, is this excessive amount of clotting. And when you get excessive amounts of clotting in the body, basically that means blood flow is blocked to many different organs. It can be blood flow to the heart can be blocked, blood flow to the kidney can be blocked. And what we're starting to see is blood flow to the brain is being blocked, and that's called a stroke. So we started seeing it in the younger kids first. They weren't getting a lung infection, but they were starting to get these problems with clotting. So some of the earliest presentations of COVID-19 in kids or young adults was actually stroke. Now you're dealing with somebody who's had a stroke, and you're dealing with the sequela of that, like a stroke from any cause. How do you protect the brain? How do you rehab the brain and things of that sort? Fortunately, the kidney can come back. Sometimes the heart can compensate. And it looks like this massive inflammatory reaction, this multisystemic inflammatory reaction that the kids have, probably is due to a combination of things going on with their blood vessels and an excessive inflammatory response that now has its own name, multisystemic disorder, but is very similar to what we used to call, what we still call is Kawasaki's, which was the result of a particular kind of virus. Those kids do seem to recover, though we're going to have to watch their hearts if we in fact give them gamma globulin and good supportive care. But I agree with Sumit that probably if we can clear the virus, we'll clear the virus. But you know, it's like a tornado. It's the devastation that it leaves behind that becomes the real issue.
Thank you. We have another question in the chat. Have anti-clotting meds be added to treatment of COVID-19?
Yes. Yeah. So now giving patients heparin is part of the routine treatment. Though, as Sumit said, this is so rapidly evolving, not just scientifically, but even in medical management. Just how one runs a ventilator. When in January and February, in running a ventilator for these patients, one thought one had to do pressure and rate in different ways, and then all of a sudden that changed to a different strategy. So starting to add heparin, which is an anti-clotting factor, has now become routine for these patients, but that's routine as of 2 months ago. Simply because there was the observation that the patients that were on heparin seemed to do a little bit better than the patients that weren't. But that is routine now.
Thank you. We keep hearing a vaccine may be ready by early 2021. When do you realistically expect a vaccine to be available? And once it is, what is the standard time frame for getting enough quantities to the public?
Yeah, 2021 is, I would say, a very optimistic timeline. You know, in the past, getting a vaccine in 5 years has been considered amazingly fast. Okay, so this is an incredibly ambitious goal here. So fortunately, we're not starting from scratch. So there were efforts when the first coronavirus came out in 2002, 2003. There was a campaign to start different vaccines to treat that. So unfortunately, when that virus went away, the funding for those studies went away. And you know, there was a story that a colleague of mine, you know, was going to develop a clinical trial for their vaccine. It cost $40 million, and everyone said, "Hey, no, the virus is gone. We don't need it anymore." But $40 million sounds like an amazing bargain right now, I think, for having a tested vaccine in place, right? So I think one thing that we should learn is that, you never let your foot off the gas on these scientific endeavors, even if it doesn't seem like a threat today. We need to keep pushing and making sure that we have everything in place that we could possibly have, because you never know what's going to happen. So getting back to the 2021 question, I think that that's if everything goes perfectly. And I've been in science long enough to know that things don't always go perfectly, and they go wrong more times than they go right. So I would say that, probably a 2 to 3 year time frame is more realistic. That being said, there's never been such a concerted effort worldwide from academics and from industry to develop a vaccine. So I'm happy to be wrong on that. And then the second point is manufacturing, right? So bringing these to scale and really you have to vaccinate the entire world population, okay? So there's manufacturing, there's compliance issues. We've seen here, even in the US, right, where we have good vaccines for measles, people aren't getting them, right? So there's a number of logistical things that need to be put into place to get to what's called herd immunity. That's where somewhere around 70% of the population has it, has protective immunity. And understanding, getting back to a question you asked earlier, understanding how many booster shots you have to take, how long that immunity lasts. This is going to be a challenge for years to come. But I think that we should be optimistic for 2021, but I think realistic for 2022, 2023.
Thank you. How do you understand lack of respiratory distress with very low PO2?
It's clearly a question from a physician. So most people can so PO2 is the amount of oxygen that you have in your blood. And red blood, arterial blood, has a high oxygen level, and venous or blue blood has a low oxygen level. And the way that you take blue blood and turn it into red blood is that the blood has to go through the lungs. And the lungs capture air in the air sacs. They're called alveoli, and the blood comes very, very close to these oxygen-filled air sacs and just sucks up the oxygen. So a low PO2, low oxygen in the venous blood turns into high oxygen, and ultimately what goes out to the body as arterial blood. If the air sacs are filled with fluid or they can't open up, there's not a lot of oxygen there, so the oxygen level never gets boosted. In most situations, even with COVID-19, if your oxygen level is low, you will have respiratory distress. That typically happens. So we can tolerate some low PO2s, low oxygen level, if you're just used to it. So certain kids who have cyanotic blue congenital heart disease just learn to live with low oxygen levels. If you have chronic obstructive pulmonary disease, you learn to live with low oxygen levels. Most of the time, however, when it's low, you do have respiratory distress in some fashion. You breathe quicker, or you breathe more deeply, or something. Now, in the cases where the level is low, but maybe you do not see respiratory distress, it may simply be that the sensors have been blunted. The sensors are not working well. And we do know now that some of the patients that presented with COVID-19, some of their initial symptoms were not respiratory. They were actually neurologic, meaning a loss of taste and a loss of smell and that's in something called the brainstem, which was the first sense that there is a neurologic component to this disease. Now we've recognized that there's a very prominent neurologic component, but that was the first sense. So the thinking was that maybe the first part of the brain that gets hit is this brainstem, where all these nuclei are that can mediate fundamental things like smell and taste. That's also where the sensors are that respond to oxygen levels and force your diaphragm to work harder or force you to breathe more quickly or more deeply. So I think in the cases where, not as common, but in the cases where your oxygen level has lowered, but you're not seeing respiratory distress, that doesn't mean you're not going to suffer from it, but maybe your body's not responding. In those cases, it probably is that the chemosensors in the brainstem have also been blunted, and they're not giving the response that one should see in response to low oxygen. That's my best formulation.
Thank you so much, Dr. Chanda and Dr. Snyder, and more information about their research and the Stanford Burnham Prebys medical discovery can be found at sbpdiscovery.org. I put the website in the chat if you want to copy and paste that, so you can go and check that out. Thank you so much. This has been so informative, and we are so excited that San Diego is on the forefront, and we've got experts like yourselves working on it. So thank you for trying to get a vaccine for us and help understand this virus that is taking over our lives in different ways. So thank you very much.
Thank you, Melanie. It was a pleasure.
Bye-bye.
Bye-bye.
Bye.